Good morning, everyone. I'm Ray Jayawardhana, the Provost and a Professor of Physics and Astronomy at Johns Hopkins University. It is my great pleasure on behalf of all of us at Johns Hopkins to welcome you to this magnificent facility. The Hopkins Bloomberg Center, formally dedicated last October, is a new beacon for discovery, learning, and innovation at the heart of America's capital. As we celebrate Kavli Laureates today, we are also celebrating that indomitable human spirit and our collective quest to make sense of ourselves and our place in the universe. And now, without further ado, it is my pleasure to introduce the president of the Kavli Foundation, Cynthia Friend. Over to you, Cynthia.
The Kavli Prize is a signature program of the Kavli Foundation. And the foundation itself was actually established by Fred Kavli, who is a Norwegian-born American entrepreneur and businessman. And it was founded in 2000 with a vision to advance basic science for the benefit of humanity. And personally, I find that to be an absolutely remarkable vision. He was interested in supporting specific science fields, and based on his own curiosity and also consultation with scientific and academic leaders, he chose to support 4 fields of science through our philanthropy and basic research. And those are astrophysics, nanoscience, neuroscience, and theoretical physics. Fred also recognized the importance of making a strong connection between science and society so that there can be a deeper appreciation of the importance of basic science to our world and to our society. The Kavli Prize is actually a partnership with the Kavli Foundation and Norwegian partners, namely the Norwegian Academy of Letters and Sciences and the Norwegian Ministry of Research and Education. We're very grateful for their administration and participation in this event and in this selection process. It fosters connections between Fred Kavli's 2 home countries, Norway and the United States, but also it's meant to foster connections across scientists around the globe. Since the Kavli Prize was established, 65 scientists from 13 nations have already been honored, and today we're going to add to that illustrious group. I'm very excited about this because they're pioneers in their thinking, they're persistent in their pursuits, and incredibly creative individuals. I'm really excited about this announcement. And we're very fortunate to have quite a few of them with us today in person. Join me in welcoming them to the stage. We'll start with astrophysics, Professors Charbonneau and Seeger. In nanoscience, Professors Langer, Elvisados, and Merkin. And in neuroscience, we have Professor Vinrik Freiwald, who is here today in person. So please welcome him. Unfortunately, the other 2 neuroscience laureates, Professors Kanwisher and Zhao, are not able to join us today, but we're looking forward to them joining us in Oslo. We're also very lucky to have a longtime partner, Frank Sesno, who will be moderating this discussion. So please join me in welcoming Frank Sesno to the stage.
Well, thank you very much, and good morning, everybody. And good morning to all of you. Congratulations. Should we do one more round of applause for the laureate? It's a great pleasure and privilege to be with you and to acknowledge your work and talk about it and you and where we go today. So we'll do that in the time ahead. A little bit about what it was like to win this prize, what it means to you. We'll hear some more about the work that you're doing, the science, and maybe a little bit about your origin story, like what motivated you to take this on to begin with. So, Dave, if we can start with you coming down the line here, how'd you learn about this prize and winning this, and Yeah, right. You get to all grab the mics here because you'll use those. What was the moment?
Well, the moment was very late at night because I was in New Zealand at a scientific conference, and the Kavli Foundation had invited me to join a Zoom call because they thought I might want to give a lecture as part of a colloquium series. And I thought, well, that sounds great. But of course, they were in Norway, and I was in New Zealand at this meeting. And so the only time that worked is it would be midnight for me. But then the moment I got on the call, I saw my colleague, Sarah Seeger, and we heard the wonderful news, and I was just elated.
Were you surprised by the news?
I was completely
how
surprised,
did-
absolutely surprised.
We
by
have
the
a
news.
we have a secret to tell you here. I mean, how was this delivered?
Oh, it was, I think, the first sentence that they told us was that, actually, we've brought you here today to tell you that you are the laureates for 2024.
Sarah?
I have a personal set of values I live by, as we all do, and one is to never, ever lie. And they had said, hey, would you be interested in helping us organize a symposium? But I guess in some cases, white lies are okay. So we had a bit of trouble coordinating because they wanted us both on the call. And it was 4 a.m. my time because I was on the way back from British Columbia, Canada. And I was getting a ride.
4 a.m.?
Because I was getting a ride to Spokane. So I was in Spokane, Washington, getting ready to get on my flight. But I wanted to get the call done so yeah, I was super surprised as well, and it was really early, pre-coffee, so it was hard to process.
What did somebody say? I mean, when you got the call?
Well, as soon as we got on, it
Congratulations.
was suspicious because it wasn't us and the person who wanted us to organize a symposium. It was a lot of people. So I knew something was up, but I didn't know what.
Now, you have, both of you, a lot of accomplishments and a lot of prizes. Is this still an important thing to you?
It's big.
Yeah, this is a huge deal for sure. I honestly never thought I would win the Kavli Prize, and when I started working in the field of exoplanets many years ago, this was not something that was even remotely on the horizon. So it's a really profound honor.
I'd like to say it's a big deal for the field of exoplanets. When we first started working on exoplanets, people thought it would dead end. And even when it started to pick up, we were accused, as we often are in astronomy, of stamp collecting, just collecting things that will go nowhere. Today, we're getting ready to work on NASA's Habitable Worlds Observatory. And so for exoplanets to get the Kavli Prize for the first time, it's good news for our whole field.
We're going to come back to that, the whole idea of being out there on the front lines, on the frontiers of your field, because you've all done that. So, Vinrik, how about you? How'd you learn about this, and what does it mean?
Very similar story. I was completely dumbfounded before we got this decoy of organizing a conference that worked perfectly well for me. I had like zero suspicion. I was actually very excited, maybe going to Oslo, organizing this conference. And otherwise, it was just like, an ordinary day. I was not in New Zealand, not catching a flight, middle of the day, with this conference call with Norway, and thinking about like how we would organize like a nice symposium. And so then, yes, first sentence of a suspiciously different group from the one that asked us to be there, telling us that we won this prize, and it was overwhelming. I think I don't know if it's really fully settled in my mind yet that this is a reality. But it certainly didn't fully, I didn't fully comprehend it during the entire
It hasn't
Zoom
settled
meeting.
in your
Yeah,
mind that it's a reality, really?
yeah, it's an amazing honor.
Does it change what you think you're going to do or how you talk about what you've done all your life to have this honor?
I don't think so. I think it gives opportunities, for public outreach, and in that sense, it'll change, how I think and how I talk about my science.
Sure.
But I think we all have a compass of what we want to do, and we'll follow that.
Bob, how about you?
Well, I'm trying to remember. I was at a restaurant with my wife and some friends, and I get this text message. But it wasn't that you got the award. I'm trying to remember it exactly.
You tipped him off?
No, but it mentioned both. Then it said, well, maybe we could have a call in a week with Paul and Chad. So I emailed Chad. I said, do you know what this is all about? So
it's pretty fun. No, it's exactly. I was at dinner and I hadn't looked at my email, and I get a text that says, we're trying to get a hold of you. We want to set up a Zoom call, and I got off it, and they said they got my text message number from a colleague, and I got off and I looked at my wife and I said, I think I won the Kavli Prize. Hold on. So then I went back and looked through my email because they said there was an email, and then they had this faux email, and that's when I texted Bob and I said, "Do you know what this is all about?" He goes, "No, I'm not sure." I said, "It's looking pretty good because Paul's in there, you're in there, and I'm in there."
Oh man!
What'd your wife say?
I just rolled her eyes.
Okay, now so look, I mean, the subterfuge was pretty good. You're not going to be able to use this next time, presumably, because now people are going to know. But I'm sure you're creative people who come up with something else. But I'm wearing a university president hat, and scientist hat. I'm trying to do this back and forth, and so I was wearing my university hat when this thing came in. And I'm, most of the time when stuff comes in saying, "Hey, do you want to do a symposium or something?" It's like, well, I've got certain other things that are taking over, my mind right now. But then I see, it's Langer and Merkin, and I go, okay, yeah, why not say no to that? I mean, these are the people who do the beautiful things. So you can't say no to that. And I thought we were just going to do literally, I really thought we were just going to do we're going to do a symposium type of thing. So it was a good subterfuge. It worked.
I think you may have your next field of research, too, is to figure out what part of the brain recognizes prizes. Let's talk now about your actual science, your work, your breakthrough, your contribution to knowledge and humanity. And I'd like to start, Sarah and Dave, with you. This incredible work you've done on exoplanets and this idea, and we saw it in the video, of maybe 100 billion planets out there, and where this field is. And also, maybe you could start a little bit by talking about what you've pioneered and how we have even detected these things, and provide some background on it.
Should I go first? Sure. Okay. So I showed up at grad school in astronomy, and I was certain I was going to study actually a completely different field. I was going to study cosmology, which at the time was clearly the focus of astronomy. It was quite a mature field. And then I went to a great talk by a professor, Bob Noyes, who would later become my advisor, and he said, actually, someone's just found a planet orbiting another star. There's a big debate about whether it really is even a planet. Maybe the signal's being misinterpreted. And that just seemed like a thrilling opportunity. It was such a practical question. Is there really a planet or not? It was clear that it would lead, it was very easy to come up with questions that were relevant that nobody knew the answer to. How common are these planets? What are they made of? What are their atmospheres made of? And so I tried a first project, and it didn't really work. And then for my second project, Bob was a great advisor because he said, actually, you shouldn't keep working with me here. You should go out to Colorado and work with this other guy, Tim Brown. And you're going to remain a Harvard student, but you're going to be out working there on this idea that Tim is working on about trying to find planets by a different technique, which is when they pass in front of their stars. Mm-hmm. And we call that a transit. And so I remember I bought a cheap car, I bought a Ford, and I drove from Boston out to Colorado. And the project was in a plywood shed in a parking lot. And it's a 4-inch telescope, okay? So this is a smaller telescope than most amateurs would have in their backyard, okay? And yet, with that telescope, we were able to measure the first passage of one of these planets in front of a star.
How did you do that?
Well, so basically you just have to measure the brightness very, 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 very precisely. And I think Tim's initial vision was that we would do a survey. We would just look at a very large numbers of stars. But I said, well, I'd never done this kind of measurement before, so I wanted to start with just a handful of stars where we suspected there was a planet by a different technique, a wobble method. Okay, so if the planet and star are orbiting each other, then we see the star, moving back and forth. And so I talked to some colleagues, and they suggested one particular star, which is HD 209458. It's a terribly boring catalog name, but a very important star. And we had to really fight through a lot of terrible weather. And it's a very small signal. It's only about 1% of the light gets blocked by the planet because the planet is so small compared to the star. But nonetheless, there it was. And then instantly, this proved that those wobble signals really were due to planets. They weren't some misunderstood form of stellar variability or pulsation. And right away, my favorite part of that paper is at the end of the paper. It's a very short paper. It's a 4-page paper because we wanted to be fast. And at the end, we wrote down ideas. And the first idea was, now we could actually study the atmosphere, because we had a way of working on that when they passed in front of the star. And so then that was the next step.
And you knew you were onto something. Sarah?
I was a graduate student at Harvard in the mid-1990s, a couple of years ahead of Dave. And I'd finished my master's level project solving an important problem in cosmology. But that project kind of dead-ended. And my wonderful advisor, one of the best advisors ever, Professor Dimitar Saslov at Harvard, said, hey, these brand new exoplanets have been discovered. Why don't you work on their atmospheres? Well, I was his first student, and he didn't know. And he never told me till like a couple decades later that you're not supposed to give it. Let's say you're just not supposed to give a student such a risky project. Why was it risky? Because a large majority of astronomers were pushing back that these were not planets. We can maybe get into that later, but that the signal was due to something else. Nonetheless, I did my thesis on exoplanet atmospheres. I was a newly minted PhD starting my postdoc. I, and maybe the few others that cared, knew there was going to be a transiting planet any day. And because I had the idea that he mentioned, but wrote down this idea, how you would do it, what you would look at, what you would search for, laid down this theoretical idea of transmission spectroscopy. Then I heard from Dave one weekend he had seen something, and I had to drop everything to get that paper done. Now, because the field was wide open, people still didn't take it seriously. Even once we got this done, got the paper, did the proposal, found the first atmosphere, people still weren't hiring in exoplanets.
Yeah, they said it's a one-off.
Yeah, one-off.
It's not going to turn into anything.
However, what's really interesting about this is it gave me time to repeat that. Other ideas on exoplanets, how to characterize them, how to study their atmospheres, all computer modeling theoretical. And so I had a lot of time to get a lot of that done for the first time because most people weren't caring. And it's all panned out wonderfully.
Yeah, and just to explain the connection. So when we thought, okay, we're going to go and try to study the atmosphere, we thought, what should we try to measure first? And it was Sarah's PhD thesis, right, that said
postdoc,
this,
it was my
or your postdoc work?
Yeah,
so look at this particular feature, which was due to sodium. Okay, it's not that the atmosphere is made of sodium, but the sodium, given the conditions of the atmosphere, would be very prominent, and it would be the easiest thing to measure, and that would be your first handle on an atomic feature. And then from there, you could go after lots of molecules. And that's what we targeted.
So this sets the stage for you to find the molecular fingerprints of atmospheres, right? Around both giant and rocky planets. So what are molecular fingerprints, and how do you dust for those?
Well, what's
They're very
amazing,
far away.
We'll just say we're using the same method we just described of transmission spectroscopy. When the planet goes in front of the star, some of the starlight shines through the atmosphere. And the planet atmosphere absorbs and puts a fingerprint on that starlight. And so there are gases like carbon monoxide, carbon dioxide. There's like a growing list of gases. And maybe perhaps why we're here is this field is flourishing. Dozens of exoplanet atmospheres are being studied. There'll be hundreds. There's several hundred people working on this field. It's all flourishing with the James Webb Space Telescope today.
Yeah, the innovation really was to turn the star from being an enemy, right? So the star is enormous, is 1 billion times brighter than the planet. So the star prevents you. If the planet was just sitting out by itself in space, we could see it. But they're next to stars, and the stars just overwhelm any light from the planet. And the innovation was by using the transit method, then the star became an ally. The star was the source of light that we would use to probe the atmosphere. And that was the innovation about that method. And then that's exactly right. So we went from this 4-inch telescope to the most powerful telescope at the time, the Hubble Space Telescope, and we're able to make a first detection of sodium. And then afterwards, there were studies of water. And then the other connection was to start to see the actual thermal emission from the planet. So not just the opacity as light's going through the planetary atmosphere, and you get the fingerprint, the molecular fingerprint, based on what wavelengths are absorbed of whatever atoms or molecules are present, but actually seeing the temperature of the planet and making maps of the planet. And that was another NASA space telescope that was absolutely essential for that, the Spitzer Space Telescope. And it was basically very sensitive at infrared wavelengths, and the planets, that's where they put out most of their light. And we could actually reconstruct the light that we measured, okay, and actually deduce maps of the planetary surfaces. Basically, as the planet rotates, okay, you see different changes in brightness, and then you can invert that data and actually calculate what the planet looks like on its surface.
I'm going to shift in a minute, but before I do, and we'll come back to you, and we heard in the video, the announced your laureate status, this reference to super-Earths. What is a super-Earth? And how likely is that, we can save this for later too, to have life?
Well, one of the most amazing thing about exoplanets is they come in all masses, all sizes, all orbits that are possible within the laws of physics and chemistry. It's super outstanding. So there's like a continuum of sizes. And there are many planets that are more massive than Earth, they're bigger than Earth. And we do kind of demarcate a planet that's a super-Earth is predominantly rocky as far as we can tell. We love super-Earths and their slightly larger cousins, mini Neptunes. Not too creative there. But because the bigger the planet is, the easier they are to study, to find and study. So they may have life, they may not have life, but we have to keep our minds open.
Yeah, in the solar system, obviously, we have no planets intermediate in size between the Earth and the ice giants, Uranus and Neptune, which are sort of 4 times the size of the Earth. And yet, what we learned as we looked out at other stars in the galaxy is actually the most common kind of planet is about twice the size of the Earth. And so there's this raging debate about what they're made of. Are they balls of water? Are they giant balls of rock? Are they balls of rock with gas on top? Are they mini Neptunes? And there's been a lot of progress, but I would say actually that's still a very active debate right now about their true nature.
Where are you on that debate?
Well, we're waiting for the data.
Okay.
Yep. But how crazy is that the most common planet out there that appears to be out there, we don't know what it is made of, how it formed, and people are trying to use new data from the web to sort through the options. But again, because before, remember, not too many people were working on this, I had a student, Leslie Rogers, who's a professor in Chicago, at Chicago University. We started working on this idea of could these, if there are such planets, what might be made of. They're very run-of-the-mill average density, and in fact, you don't know what they're made of because their density is so average. And as we were working on that, that's when Mirth found Gliese 1214b, the first in that category of the mini Neptunes.
May I just quickly ask the audience a question with a show of hands? How many of you believe there is life out there on another planet? Are they right, wrong, or do we just not have the data yet?
Well,
And for those who are live streaming, let the record show that, what would you say, 95% of the audience raised their hands?
Yeah, so actually I'm in the other group. I don't think it's a certainty that there's life on other planets in the following sense. So right, as I'm an observational astronomer. I build telescopes. I stay up at night, and for me, what matters is can we actually make the measurement, actually detect life, and showing there is life. And so it isn't enough to say, well, the universe is infinite, and therefore there must be life because there's all these planets. The question is really, is life sufficiently common and long-lived that within some reasonable volume of space that we could ever hope to study with real telescopes, that we could find another example of an inhabited world. And so then you're talking sort of 100 light years, 200 light years, and you're talking about thousands or tens of thousands of stars. And then I think it's not unreasonable to say life is either unique or such a rarity that we are the only example within that volume of space. The exciting part is that the first time you have the sensitivity to make that measurement for another planet that's roughly the right temperature and composition, right away you know what parameter space you're in, right? Maybe we live in the Star Trek universe where every star has a planet with life, and all you have to do is go measure the closest one or the closest 5, okay? If you don't find something in the first 10, then you sort of slog it out and you work your way up to 10,000.
I just want to add one thing to tie this off, that, in exoplanets, because it was such a struggle in the early days and now it's so mainstream, I love to say in exoplanets, and especially the search for life, the line between what is considered mainstream and what is considered crazy, that line is constantly shifting. So we need to inspire the next generation to think about the crazy ideas for new paradigms of space telescopes. Maybe it's self-assembly in space. It's something that we don't know how to do yet. Just maybe they'll get the Kavli Prize a few decades from now. And we need to expand that volume and do bigger things in the future.
Brilliant, brilliant. All right, let's go from the largest to the most complex. And we'll go from planetary recognition to face recognition. Remarkable work. What does it tell us? Why is it significant that a part of our brain, a designated part of our brain, is engaged in face recognition?
So the way neuroscience is presented here, and I think it's one of the greatest allures of neuroscience, is really the study of complexity, right? We have the very small, we have the very big, in between we have brains. But what really stands out about them is that they're very complex. I also want to point out that they are of very fundamental importance, right? Because everything that we can possibly know is happening because of our brains. And so they're not just an object that's in the middle of everything else. And that is, I think, one of the greatest attractions, for studying the brain. But we are struggling to understand its complexity, and there are different ways you can go about it. Corey Barkman is here with us today, who established a new model system with a simple nervous system that makes it possible to really study brain function. And we are studying the most complex brains, human brains, and the brains of human next relatives in the primate order. And they are organized at many, many different levels of organization. And so bringing together multiple techniques, we're able to reduce this complexity and make it tractable, yet at the same time embrace it, and then really study, like, why is this complexity there? What is it good for? And the answer has to be in information processing.
Could you talk a little bit about the functional magnetic resonance imaging work that you've done and how your methodology engaged that technology to identify this, what you saw, what you did?
Yeah, so this, many of you will have seen these images in popular magazines of, particular parts of the brain lighting up. This is a false color code for parts of the brain that are more active in one condition than the other. And this is really possible because of a measurement technique that does not really measure brain activity, but measures changes in blood flow or changes in oxygen that your brain needs to function. And so whenever a part of the brain is more active, performing a particular task, there's going to be more blood flowing there, more oxygen delivered, and more oxygen consumed. And that's actually what this technique is measuring. So it's an indirect measure of activity. And that's why the next step for us was really to move an electrode into this region to directly measure the brain in function and really show that the cells in this region are selectively responding to faces, and then to show how they encode faces.
So we saw in the video somebody going into the MRI, right? What's happening? Are you showing them things, and are they seeing faces while they're in there?
Yeah, exactly.
What are you tracking and discovering?
Yeah, so there's a person lying in the scanner, looking at a screen and watching a different stimuli, including faces. This is one of the conditions. Some controls that could be hands, could be bodies, could be non-biological objects. And then afterwards, you're contrasting activity at every point in the brain while you're watching faces versus when you're watching everything else or a particular control condition. And that's the way for you to find regions in the brain, if they exist, and they do exist, that are selectively responding to faces. And so it's this combination of this measurement technique at the same time presenting the stimuli, or you ask people to do something if you're interested in cognition, give them a task, and so on and so forth. So you can apply this technique very widely, and this is the revolution that really Nancy has brought forward, using this technique, combining it with cognitive psychology, and then asking the questions that you would otherwise ask your subjects to every part of your brain.
Now, in the video we saw, we heard you talking about face processing and recognition of individuals as distinct activities coming from distinct places. Could you talk a little about that and what that means and why that's significant?
Yeah, so I think that's maybe surprising to everyone because our perception of a face and our recognition of individuals, they're so closely tied that it seems to be the same process. But really one is about a part of the brain that's analyzing information that's coming from a particular sensory system, in this case from our eyes, and is trying to identify what is there. And it's doing this for a number of different things, including faces, but certainly not limited to it, any object, anything that is there. We think we're building models, to explain this reality that is out there. And that's a perceptual process involving lots of computations that we're interested in determining. But then there's a link to memory. And that might actually be the origin of the evolution of faces, is that we know people. We are social beings, we're around people that we know, and that information is stored in our memories. So the question is, how do you go from this general perceptual process to recalling particular memories that this is David, right? This is Frank. And so we discovered two brain regions that are critical for this transition from perceptual processing to activating your memory.
That's really interesting. So, Vinrik, I am proud to say that I have a 100-year-old mother-in-law. She's remarkable. But her short-term memory is gone. but she still recognizes people. So what does that tell us, and how does your research connect to that?
Yeah, so lots of our cognitive abilities decline over time. I would also say that maybe the story of decline is a bit misleading because there are reformations and changes in a lifetime, but many processes decay. We believe that because face processing is so important, a lot of real estate in the brain is really devoted to this process. And so there are multiple brain regions. And so even if there's like an overall decline or decay of capabilities, face recognition can withstand it for a very long period of time. But not always. There are heartbreaking stories of people suffering from Alzheimer's, a disease that is originating in this region that the face areas are located, also the face memory regions. And the moment this person loses its ability to recognize its loved ones, that's already a form of death for this person and for the entire family.
Okay, so I'm sure we've also all experienced this, right? We see somebody, I know you, I see you, I recognize you, but I can't place the name. Different functions, different parts of the brain. What's going on there?
Different parts of the brain, yeah. So lots of people have difficulty remembering names, I think much more than faces. There's, however, this interesting condition of face blindness. People that are not able to recognize faces, actually an early influence on me as a high school student, picking out this book from the neurologist and popular science writer that many of you know, Oliver Sacks, describing the story of a man who could not recognize his wife and acted accordingly. So, yeah.
May I invite you, since you're the sole representative of your group, to reflect on your colleagues and the contributions they've made and how you've worked together and collaborated?
Yeah, so Nancy is really the pioneer bringing fMRI to the study of complex functions, and as was explained in the video, she applied it to face recognition, and so she discovered this fusiform face area in humans, the first face selective area that we knew of that exists in every subject that she studies, and that all of you have in your brain. And that set forward this revolution in cognitive science, really allowing us to take brain measurements where before, you might have been able to ask a subject. And then Doris and I, building on this, we used fMRI to localize this region in animals in which it was now possible then to really figure out in detail, what the individual cells are doing, how they're encoding faces, and do these detailed, very mechanistic studies that ultimately, will lead to also new understanding of algorithms, like how our brains compute these functions.
All right, so we'll come back to you and some more of that. Let's go then from the most complex to the smallest, right? So to nanoscience now. And you're recognized and laureates for your pioneering work integrating synthetic nanoscale materials with biological function for biomedical applications, which means, as we heard, going to and addressing some of the diseases that we commonly have. Can you talk about the connection of the nano to the bio?
Well, I mean, I think everybody's done, I think, different things in different ways. I'll maybe start with mine, and I'm sure Paul and Chad will go over other things, including imaging and diagnostics. Ours, a lot of what we've been involved in is therapeutics and even in drug discovery. So for us, for me, it started over 50 years ago. I was doing postdoctoral work with a surgeon named Judah Folkman, and we were trying to come up with a way to study how blood vessels grew and whether we could discover molecules that could stop that from happening. And there wasn't really a good way to study it, and we needed to deliver what are called large molecule substances. So for reference, proteins and DNA and RNA are large molecule substances. So there wasn't a way to do that, and so I tried to figure out a way to do that, and of course I failed hundreds of times, but eventually we were able to make tiny particles that could do it, and that enabled us to actually discover that molecules like this did exist, and later people would use those approaches to isolate substance or to find substances like Avastin and others that have been widely used for treating cancer and diseases of blindness and so forth. And then we took it a lot further. We began examining mechanisms of how these worked, creating new materials, looking at applications. I think in the video it mentioned vaccines, and of course that took on a major thrust as the years went on. I mean, a good example, a company I helped start is Moderna, and there you're using nanoparticles, in this case lipid nanoparticles to deliver, the messenger RNA. And of course, if you didn't have that, then the messenger RNA would be destroyed. The vaccine wouldn't work. So, but there are many examples. People are using these kinds of things for treating, mental health diseases, heart disease. I mean, pretty much every kind of disease. And then if you look at some of the other things, I mean, Chad's been involved in both, therapeutics and diagnostics. Paul's been involved in a whole bunch of things. And so I should probably let them expand on all this.
And I'll let them do that too. And I want to do this somewhat methodically, talking about past, present, and future. But what you said kind of, I think, inevitably leaps us forward. Is this and this technology and this science the kind of thing that will ultimately cure cancer or eliminate some of these horrible things from human existence?
I think it will help. I think it would be wrong. It's the biggest issue, I still think some of the biggest issues with cancer and with other diseases is understanding the biology well enough, to do something. But if you are able to do that, I mean, then, and just to pick cancer, one of the things that's being worked on is to, use genetics to understand what are called the neoepitopes of the cancer cells, which are different than the regular cells. And then you can design a messenger RNA vaccine against those cancer cells. And that's now in very advanced clinical trials, phase 3 clinical trials. And, and the data have been pretty remarkable. So the hope is that by understanding these things better and better across the board, it'll help. But it's really, I think there's something that we call convergence, that if you want to try to treat a lot of these things, it really involves disciplines across the board.
Sure.
Nanoscience is one of those key disciplines, but there's others as well.
Chad?
Yeah, I would just build on that. I would say it a little more aggressively in the sense that I think it will cure certain forms of cancer. To say cure cancer is just a ridiculously broad.
But it's a breathtaking.
But I think many of these materials, look, when you take any material and shrink it down in size, it has different properties. That's what makes nano so interesting. Everything old becomes new. When you restructure molecules on this scale, they have new properties. And those properties have changed the way we track and ultimately treat disease. And in the case of cancer, there are a lot of promising experimental therapies that are out there that in clinical trials have cured certain forms of cancer in certain people. And that's the other thing. It can be population sensitive as well. But I think the unifying feature is that these structures enable new opportunities that are really advancing medicine in a significant way.
Well, let me take one step back slightly and just start with, why was Fred Kavli wanting to honor nanoscience? And one of the things that's going on is this dream is being realized of the ability to actually control a material on the exact same length scale that nature builds proteins and other structures at. And these are things, if you just look at a piece of kind of ordinary matter that people have made, I don't know, a picture of steel or something, and you look at a protein, I mean, you look at the structure of a protein and you say, oh my God, that is just unbelievable what nature is able to create, the complexity and also the precision and so on. And what's happening is work where we're learning to make artificial materials where we've controlled them really exquisitely on the same scale as the functional units of biology. Mm-hmm. And one way of proving that is to actually take those things and put them in a biological system and have them execute some kind of a function. And this is a big breakthrough for us. I mean, there's the idea of, say, an implant or something, you're sticking a macroscopic piece of material into somebody or an electrode or something. But, here we're making, a little object that's on the same scale as the objects that are naturally doing their, their controlled business inside cells and so on. And for us, that was, there's a whole story, at least for me, about what attracted me to wanting to be in that business. We can maybe talk about later. But there was a moment in time where we were working on these abilities to make tiny materials, and people were kind of saying, well, that's kind of interesting. Is it ever going to do anything? And the answer was, of course, eventually yes. And trying to get to that moment was a real journey and a fun and special one. And so, I think that's maybe a notion to leave with here is how much the beauty of the science of making things and these very small scales is connected with understanding really the beauty of biology and how those two things can be brought together into a kind of way of cooperative discovery.
It's a great way to put it. And Chad, you've talked about your work trying to figure out how to, and I think these are your words, build matter from the bottom up.
That's correct. I mean, so we weren't trying to develop anything having to do with biology or medicine. We were trying to redefine chemistry, redefine the way you make materials. And we had an idea that you could, instead of build matter with atoms, you could build with particles. And instead of using electron-based bonds, you could use short snippets of DNA as kind of a glue, a programmable glue that would then program the formation of matter from the bottom up. And there's a whole field called colloidal crystal engineering with DNA that was spawned out of that. But in the course of doing that, in fact, in the first experiment, I had a student, a guy named Bobby Music, who came down at 11 o'clock at night and said, Chad, you got to see this. I put DNA on these gold particles, which are ruby red in color. That's one of the really interesting things about miniaturization. Everything's different. Not gold that associated with your wedding ring, but intense red color. And I mix these particles that are designed to be complementary to one another. And what you see is a striking colorimetric change. It goes from red to blue. And he said, that's not the cool part. He said, here's the cool part. Put this in an oven. You go above the melting temperature of DNA. The DNA is a double helix, and it dissociates at a certain temperature, and it goes back to red. You put it back on the benchtop, and it goes back to blue. Back, forth, and almost in unison, we said, oh, my God, new form of detection, new way of detecting DNA. And just kind of kidding around and not really knowing what you would have to do to develop a detection system. But then we started saying, hey, let's really probe this and ask, are these new properties really useful in this area? And that drove us down the biodiagnostic front, and then we just kept following our nose and learning more and more about this new form of DNA that we now call spherical nucleic acids, and found out they could not only be used as biological probes to track disease, but they would, for example, go into cells, human cells, where normal DNA won't. And you could use that now to begin to manipulate what goes on in cells, and now you could begin to create shots on goal for new therapeutics. And so we've taken 7 drugs into the clinic, and many more are planned, and are trying to redefine how a big swath of medicine is ultimately practiced based upon some of these concepts.
One of the things that you were recognized for in this prize is the way you've taken your science and brought that to the marketplace through the companies you've started and other things. What is most significant from that? What are you proudest of? What's the biggest impact? Tell us how that has translated in your view.
Oh, golly. So, kind of a similar story to what Chad was just describing. For a variety of reasons, I was extremely interested in controlling these materials. They had very beautiful optical properties. And I was in this milieu where, in this case now, at Lawrence Berkeley Lab, where there was a lot of work going on to try to really image cells at tiny scales, see single molecules and so on. So in some, sitting with a colleague there, Shimon Weiss, who was one of the very early single molecule spectroscopists, he was telling me about how all of his materials that he was trying to image kept fading. It's like, well, heck, we can put a quantum dot in there, and that's not going to fade, ever. And so then there was this, wonderful student, Marcel Boucher, and it went pretty quick, to when, okay, we thought, gosh, let's go do this, to, like we did it by making certain coatings and so on, which were quite important for this, and are related to coatings that are widely used today in different ways. But let's just say pretty early on we got to that and could publish an early work just saying, you can take these materials and put them inside cells, and they'll go and decorate very, very precise spots if you do the chemistry right, and then you can image what's going on inside there, and guess what? They don't fade, and you can have lots of them and look at different colors and so on. And, at that point, at least for me, I had this really strong feeling that I was not the person who would know all the things for to do with these. So I was determined that we would make this commercially available. And I think, maybe the first paper came out maybe late 1998, and by 2000, there were 1,000 labs that had these materials in their hands to do work with them. That's how clear the need was. This had become a commercial product that could then be used. And their demand for it was absolutely enormous. And of course, for example, they're used for discovery all the time, for cell tracking, for all kinds of things like that. But especially they're also used very, very frequently in pathology labs to look at biopsies and so on, and to try to tell these, malign or malignant or benign growths and so on. So they're just used all over the place, and for me that was extremely satisfying. The idea that we would make a material and then it would turn out that people could use it for such beneficial uses was a kind of dream.
Jan, what about you?
Well, we took a kind of a proactive strategy. My view was we were there in the early days of what I call the modern era of nanoscience, and I thought for this to have impact that we have to get it in the hands of the public. We have to do the translation, and so I began to work with a lot of folks that could help me build these companies. Befriended Bob, who's the king of doing this.
Moderna, we've heard of that, right?
Not just Moderna. He's got a lot more than that I'll tell you about. But you look at people that do this really, really well. You can learn a lot from them, and I learned a ton from him, and kind of followed much of his.
What's most significant to you that this has contributed? What are you proudest of?
Well, as I said, these constructs, which constitute a fundamental new form of, I think, the most important molecule ever synthesized by chemists, now have enabled us to track and treat disease in new ways, and are saving lives and improving lives.
Yep. Briefly?
Well, I think maybe also to take a step back. Why do we start these companies? To me, it's what Chad said, and Paul. You want to get these things out to the public to help people, to improve lives, to save lives, and for me, I was very naive when I did things in the 70s and published papers. I just thought you publish it, and people will use it, and we even patent things. But still, I did get calls starting in the 80s from some very, very large companies. And they said, well, they do want to use some of these things, and they'll license things. They'll even give us a grant, all of which they did. But then, after a year or two, they just gave up. So, one of my friends, Alex Klebanov, said, well, Bob, we should start our own company. And we did with some of my students. And like Chad mentioned, we've kept doing it. If you take Moderna, if you really look at what's happened, and it's not just Moderna, but, you look at the United States, you look at the whole world, there's all these large companies. No large company ever took the risk in doing messenger RNA and nanoparticles until it was Moderna and BioNTech in the United States, BioNTech in Germany. Finally, in 2020, Pfizer did something with BioNTech. But if you don't do these things yourself, they're just not going to happen. And it saves millions, maybe billions of lives.
I want to come back to astrophysics now, and I want to play a video clip. So there were juries that selected these winners. And we have some of the jurors with us. And I'd like you to hear what Thomas Henning has to say about your work.
We actually embarked on a big journey. We discovered one planet many years ago, and then we now have about 6,000 planets discovered. But the real question is, what are the properties of these planets? So David Charbonneau and Sarah Seeger, they really embarked on this journey and laid the foundation for the characterization of these planets and their atmospheres. And of course, the next big step is to find evidence for life on other planets. This is a very complicated journey, but Sarah Seeger and David Charbonneau really made the steps in that direction. And I'm very convinced that in a couple of years from now, you will see the success of this foundation.
Wow. In a couple of years from now, we're going to see success. What are we going to see in a couple of years? What's he talking about?
Well, until now, as he said, we have thousands of known exoplanets, but we don't know much about a lot of them. We have their average densities. we're hoping and planning, and not we is several hundred people, to use the atmospheres to try to get information about the interior and physical processes on the inside, what the planets are made of, how they may have evolved, maybe even how they formed.
Yeah, I mean, so for many years, the study of exoplanets was the study of Jupiter-like gas giants because they were bigger and they were easier to access. And the big pivot that's happened really in the last 5 years is terrestrial worlds. We now know, we can point at nearby stars, we can say there really is a planet, it's the same size as the Earth, it's made of rock and iron, we've measured the density, we know how far it is from its star, and we know it has the same temperature as the Earth, and so we can set about trying to study the atmosphere. And I would say the immediate question is, do those planets even have atmospheres? We should not assume that things went down the way they did here. This is just one example of chemistry and physics over 4.5 billion years. And a question that I think we will answer in the next year or two is simply, can we show that there really are Earth-like atmospheres, or even atmospheres of any kind, around some of these rocky worlds? Even before we get to the point of asking, are there biomarkers? That's great, but just showing there is an atmosphere that's got some carbon dioxide or some nitrogen.
Right.
Just think about this for a moment. Our planet Earth, a haven for life. Think about Venus, so hot due to massive carbon dioxide greenhouse atmosphere, although the atmosphere is the right temperature for life. Will we find that so many planets out there are more like Earth, or are they all going to be like Venus, or some other options we haven't thought about yet? So that's what Thomas is referring to.
What are the technical challenges to getting there?
Planets are small and faint. That is the challenge, okay? So the stars are big and bright, and they obscure our view of these planets. And so what we need is, we need very large telescopes because we need to gather a lot of light because it's a very feeble signal. And we need to have sensitivity at infrared wavelengths. And so a major advance there is the NASA James Webb Space Telescope. That is much larger, more powerful than the Hubble, but it's also sensitive to different colors, different wavelengths of light. Molecules are mostly active, spectroscopically active in the infrared and not the optical. That's the other big advantage. But then looking ahead, I think I do want to point out an important part of this story, which is a lot of these planets that we hope to study, they orbit a very different kind of star, small stars, which we call M dwarfs or red dwarfs. And that's been a big focus of my research, is finding those. But we do want to make sure we're also studying Earth-like planets around sun-like stars, because maybe there's something about the star itself that allows life to flourish, and those other stars, even though they're extremely common, don't allow life to take root.
So let's come back to neuroscience for a minute and go to a juror there, and John O'Keefe and his thoughts about what he saw and learned from you.
One of the fantastic things that the brain does is it takes the image on the retina, the visual image, and it dissects it into a whole series of representations of different kinds of objects, trees, houses, and it sets them all within a spatial framework. One of the most important features of the environment that it represents are faces. We're social animals, and representation of faces essentially is the gateway into our whole social interaction with each other. We need to know whether what we're seeing are the faces of friends, of foes, whether they're familiar or unfamiliar faces. And this is what the Laureates have done for us. They have shown us how the brain does this, how it takes faces seen as a picture or a snapshot and puts several of those together and creates the overall representation of a face. And we think this is not only telling us about faces, but it's also telling us about how other objects might be analyzed as well. So it's a fantastic discovery, not only about faces, but also about how the brain represents objects in general.
Vinrik, back to you. I mean, fantastic discovery. It's great. He talks about us as social animals. And here's what struck me. This is essentially the gateway into our whole social interaction with each other.
Yeah, I agree. So faces are an object that we can see like other objects. You mentioned trees and others. And so one of the implications of our work is that we elucidate general processes of sensory information processing. But the flip side is that faces are really powerful agents in the social world. They manipulate us. You see a baby smile, it makes you feel warm and fuzzy, and there's no defense against it, right? We are wired this way that we have to have this response. Faces attract attention, gaze directs it away. We talked about memory systems that automatically activated, and so that's an important line of my research going forward, is to really use faces to get to these very deep, very complex processes that really define us as in humans, and then also to elucidate the human condition and the many forms that can be debilitating in disease.
There's a face I recognize, but it's in the audience here. It's another one of our jurors. May I borrow your microphone? So I'm going to do something that, it's good to come down to the audience here. And Naomi Hollis is with us. She's one of the jurors from the nanoscience, or the nanoscience team. Let me turn you, here, come on over here so everybody can see you, and turn around, and welcome. And why don't you share your thoughts from what you judged and the significance of what these scientists have done and achieved?
Absolutely, Frank. Well, nanoscience is vast, right? Nanoscience has impacted, in a broadly speaking, it can impact our ability to clean up our environment. It can impact our ability to develop new ways of doing clean energy. It can impact so many different things. But I think the chairperson, Professor Holst, said it really very, very well. The noblest goal that we can really think about is ways that we could save lives and ways that we can alleviate human suffering. And that's why this focus of nanomedicine is so very, very important, not just because of the great pioneering work that our laureates have done, but also for the tremendous potential. We really have not tapped yet into the true potential of nanomedicine. And so I think that's something we can all look forward to. We can certainly look forward to our agencies like the Food and Drug Administration being much more open-minded towards the new innovations that are coming online that have great data that shows that there are tremendous futures in therapeutics.
And their work has been breakthrough work.
Their work they are really the pioneers. They're really the ones who stood out before others and did some things, and they were lonely, in many ways, and people sort of scratched their heads and looked at what they did. But then again, many people jumped on it and said, well, no matter what I'm doing now, next week I'm going to start doing what they're doing.
Great. Thank you very much. So, and that is a great segue into the next part of this conversation about being pioneers and being lonely. You've all talked about that when we've had these conversations in the past about what it's been like to be on there's your microphone back. You get to use that again. Risk takers, lonely, pioneers. I mean, was it like you talked about? People kind of said, "Nah," when you first started.
I remember. Do you remember the conference? There was a conference that Sarah and I both attended in Corsica. I think it was a NATO science conference, and it was tiny. There was probably 50 individuals there, mostly students, and even there, it was a meeting on exoplanets. And I remember talking about a different idea about how to get at the properties of exoplanets. And but it was the same idea of trying to measure that faint signal. And the organizer laughed and said, yeah, she laughed. She didn't mean it in a bad way. She just was, she said, and she had a very distinctive French accent, and she said, but David, you will never be able to detect such a thing. And she wasn't being mean. Like I really enjoyed the fact that she organized this meeting, but there was such a skepticism that such faint signals could be seen at the time. And yet I wouldn't say I felt lonely. I would say there was like this wonderful sense of camaraderie among this group of students. And at that meeting, it isn't just that there were many other individuals that came out of that meeting and now are very important players in exoplanets.
I've always been a risk taker, but at the same time, I wasn't committed to a career in science. So I really didn't have much to lose in grad school. Nonetheless, even a professor on my own PhD thesis committee was wondering why I was studying exoplanet atmospheres. Why was I modeling clouds in these exoplanet atmospheres? Now there are entire theses on clouds in exoplanet atmospheres. And it continued as I was looking for a job. Carnegie was very forward-thinking and hiring me, but other places, I won't name them, of course, but I remember one professor just saying, we'll never have very many transiting planets. Now there's thousands of transiting planets. The field has completely exploded, and it's all good.
And you face this kind of skepticism, question, being on the front line?
Totally. I mean, okay, so just a couple of anecdotes. The same playbook here. So just a couple of anecdotes. When I was interviewing, to become an assistant professor, the most common question I got is, what would I do when the field I was working on petered out completely because it was going nowhere? And in fact, I went bankrupt as an assistant professor, because I just couldn't get any grant money. Yeah, I did. I had to go back to my department and just say, I got no grants. And in fact, actually, they said, well, you've written these really good papers, and they actually promoted me because they said, okay, and it was after
Someone
that.
believed in you.
Yeah, actually, they
really
did.
important.
They did, actually. There were people who did and who took the risk on it. But it was a very early area in the field. I mean, subsequently, I want to say that, tremendous support from many places, including Department of Energy especially. But, there was that period. And the conference thing just really rings an echo to me because there was, again, somebody from another field who thought, this stuff looks kind of interesting. And so they had this very clever strategy. There was a big meeting, and they interleaved us two different fields. So they'd have somebody from this tiny, tiny field that I was in, and then somebody from the other field, and they would have alternating talks. And there was an auditorium that would fit about 400 people, and so it would be like packed for the other people. And then when one of, our little group would come up, everybody would leave the room to go talk about it, and they'd come back in for the next one.
Please note
So
that nobody is leaving
all
the room.
all the signals were there, like, what are you guys doing? You're off in some stupid corner, But, look, I mean, I think like what you're describing, for me, I just want to share one little moment that kind of gets the sense of what these things are like. I was in an undergraduate physical chemistry lab at Chicago, and the experiment that we were doing would look at the growth of a little wire under an electric field, and very thin wire, it would just grow. And as it grew, it would emit electrons under a field, so you could see it getting brighter. You could calculate the rate it grew at, and you could show that an atom would hit anywhere on this thing and then migrate to the end, and that's how it got longer. And I just, I'm sitting in the library as an undergrad, and I'm thinking, wow, somebody could see how the atom moved on this thing, and it went to a certain place. And I just fell in love with that idea. So it didn't matter to me. I mean, what you were just saying a moment ago, I didn't really care. I mean, if I didn't get my tenure job, I was going to go do something else, It just really didn't matter. It did, but, it might not have. And then that would have, but the point was, and this is just one last thing I want to say about this, because I think it's very important. The stories you're hearing, I mean, we're talking about the kind of these aspects of sticking with something because you've fallen in love with it, because you care, because it's what you believe in. But I also just, I'm an institutionalist, and I just want to say, the universities, the national labs, the places that support this kind of work, they support people every day who are doing things that are equally crazy and that are probably, some of them aren't going to work out for sure. But some of them really do. And I just want to, I think celebrating that aspect of these are communities where people foster each other on these journeys of risk and say, okay, we're going to help you to do this. That to me is what stands out.
How about your journey of risk? Yeah.
Yeah, so I wouldn't call it necessarily risk. I didn't look at it as loneliness. I looked at it as a challenge and opportunity. I mean, I was trained as a chemist, a traditional chemist. But you could see in the 90s that there was a real interest in looking at the consequences of miniaturization, coming up with ways of making things small. This is before what I call the modern, the advent of the modern era of nanotechnology. Mm-hmm. And we decided at that time to try to build the world's largest institute in this particular area, something called the IIN, and we've succeeded in doing that, which that focuses on pure nanotech type discoveries. But I think we all have had a similar experience in that regard. I think everybody thinks that scientists are very open-minded and embrace new things. They actually don't. Most of them are very cynical, and when something new comes along, they come up with all sorts, and they should, by the way. They come up with reasons why it won't work or why this can't happen, as you experienced. But I think that's part of the whole process, and if you power through it, big things happen. So Paul left out. It was an empty room, but 5 years later, filled a room of thousands because people got really excited about it. So to me, that's part of the whole journey. I'll give one anecdote. In my fourth year, I applied for an NSF grant. I had won all sorts of what we call beauty contest money, philanthropic organizations like the Kavli Foundation giving you money for promise, but striking out across the board in terms of regular grants. And I get a review that goes like this, and I hope the person's listening. It says, Merkin's been at Northwestern for 4 years. He's done nothing of import. I don't see why the federal government should waste another dollar on his work. And I took that review and posted it outside my door, and I said to every student, I go, this is what makes me come in every day, to prove people like this wrong.
That's great. It makes me think of Marie Curie's line, right? My strength lies in my tenacity, right? You got to be tenacious, and you got to stay with it. Lowest moments, most loneliest, I mean, did you face this?
Yeah, so the stories of saying they sound very familiar. I just want to point out that there's a flip side to it, which is a happy thing, right? Is that we are following our own curiosity, we can develop our own ideas. I think you could see it in Paul when he was talking about it, right? He was happy, right? So there's a happy side to what we can do. And I think all of us here on the stage, we're guilty of exercising our rights of freedom for 20 plus years, and we should be very happy about this, that we can do this in a society that supports it and that is built on the same principle that makes this possible for us.
The connection you make to freedom is a very interesting one. Do you want to talk about that for just another second?
Yeah, so
where you're coming from on this?
I'm coming from Germany, and so we are celebrating 300 years of the birth of the great Enlightenment philosopher Immanuel Kant this year, and his motto was, have the courage to use your own way of thinking. And this, I think, is what we've all been doing, and this is all what we've been able to do in a society that's built on the same principles. This country has founding fathers, 2 of which were eminent scientists in their day. I think there's not a coincidence that there is a relation between these 2 things.
I want to ask you to address this too, but I also want, and we're coming in for a landing here because we're almost out of time, but I do want to close with each of you reflecting on a personal moment or where you came from in all of this. And so I'll combine that with you because I know, Bob, that you have a favorite poem. And it's, or at least you've talked about this, Robert Frost's poem, The Road Not Taken, that you connect with. How has that motivated you, tied into this experience?
Well, so I'm a chemical engineer and I got my degree in 1974, so almost everybody then went into the oil industry, and I thought I'd do that too. Because that's what everybody else did. They paid a lot of money, and I did. I got 20 job offers from oil companies, 4 from Exxon actually, but I wasn't very good. But it didn't really matter. But
I'm not sure that's quite true.
But I really was. Most of what is like increasing the yield of certain chemicals by small percentages. So I had helped start a school for disadvantaged high school kids, and I started looking for teaching jobs, doing chemistry education. That's one of the things I did there. And there I wrote to about 40 places, nobody would hire me. So I started, or even write me back. So then I thought about, well, how else could I use my chemical engineering background to help people? So I thought about medicine. So I wrote to a lot of hospitals and medical schools, and they didn't write me back either. But then one of the people, and I alluded to this a little earlier, mentioned to me there was this surgeon in Boston named Judah Folkman, and they said sometimes he hired unusual people. They thought very highly of Dr. Folkman. I won't say what they thought about me. But at any rate, I went there. I was the only engineer in the hospital. But I just learned so much, and it gave me so many ideas about how you could apply, chemistry and engineering to medical things. And so that changed my life.
Wow. Sarah, as I understand it, your first encounter with the dark night sky was when you were 10 years old, and it made an impression.
Yeah, I'll never forget that moment. I was a city girl, but a summer babysitter encouraged my dad for us to go on our first camping trip ever, and we went north of the city. This was in Ontario, and saw the dark night sky, and it was just wow. I hope all of you have seen the dark night sky. It's just astonishing. And I was so shocked. I was just, like, my heart would have stopped. And then
At 10 years old?
Yes, and then I was wondering, why didn't anyone ever tell me about how great this was? I've been told about other great things, but it just always stayed with me my whole life. And I just want to add something to that, because I'm sure that many people share this story. I was that very awkward child with absolutely no friends, save for my best friend, my sister. I was always wired differently. And I just want to, thank all the people. It's just what Bob said about where he was hired, and it's your micro communities that let you be who you are.
Chad, you were formed to a large extent, from what I understand, by your youth in Asia and what you experienced there. And
Yeah, I grew up in a family with a father who really never figured out what he wanted to do in life, so we were constantly moving. He joined the Peace Corps and brought a family of 6 to Korea and then Malaysia, and then came back and moved all over the world. I actually picked up another girl along the way, so we had a family of 7 when we came back. And I think that experience really made me, embrace people. So I have an incredibly diverse research group. We've got 70 people, but I think 13 to 14 different countries represented at any given time.
Really?
And to me, this is all about talent, right? A lot of talented people here, but there are even more talented people that are coming into our labs on a regular basis. We have a really privileged existence in that regard, and these people come from all walks of life and bring their talents and do some pretty spectacular things. And the ability to embrace those people and connect with them has been a big part of my success.
A big motivator for you, Paul, is connecting technology and the benefits of technology to benefit humanity, right?
Mm-hmm.
Where did that come from? Did you have a 10-year-old night sky experience, or did you grow up someplace?
All right. Well, if we're going to go, okay, we could go there. So, and coming back to the freedom topic. So, when I was 9 years old, my mother died, and my dad wasn't able to raise us. So we were sent back to Greece, where the family came from, and we were raised there by like the collective family. And that was in the middle of a dictatorship. There was a dictatorship at that time, and then eventually it fell, and it was replaced by a democracy. And the one thing I wanted to do was to come back to the freedom in the United States, because I just felt that. And when I was an undergrad, that felt to me, I was at the University of Chicago, anything could be questioned. And it was a moment where I could find something that I thought was beautiful. And that's what I think really connected. And just one more quick thing on this, to what Naomi was saying, that search for me, for beauty is kind of wanting to be able to make things that are as intricate as what nature makes. I still think we're going to make some much better nanocrystals that will do many more things that will be, of importance. But this idea that somehow the connections between discovery and freedom. I really want to echo that and say that also the places that allow for that are the places where we will see humanity thriving.
Vinrik, we're coming down to the wire here, but I was struck, and as I was reading about you and listening to you, about the high school in Oldenburg you went to that was more than 400 years old, and what that meant.
Yeah, so I think my high school really is the place where all the questions that I'm following, all the things I'm interested in, really those developed during this time. And I think the most specific thing is, thinking, okay, if everything that we know about physics, chemistry, biology is true, then who am I? And that's kind of what got me into science, and that's what got me into neuroscience and, trying to figure out the human condition.
Dave, I've saved the loftiest for last.
Oh. Okay.
Star Trek and wallpaper.
Oh. How did you find? All right. So, I was the kid that just was in love with science fiction. This was I was growing up in the 1980s, and so I watched Star Trek and I watched Star Wars. And then, of course, I was so inspired by the shuttle program, the NASA program. And they had just invented this new kind of wallpaper where you could actually have a photo instead of just having a pattern of ducks or whatever. You could have a photo, and I begged and begged my parents to let me have this photo of the space shuttle in front of the blue marble. And I really did. I just sat there and looked at the thing all night and fell asleep and would wake up looking at the thing. And of course, what's so interesting is, we don't have spaceships yet that allow us to go to other stars, but that's just not when I'm alive, okay? But astronomy and telescopes are sort of like a spaceship, right? You get a group of people together, you got your team, just like Captain Kirk's got his team, and you can go out and explore the universe, but using these very different kind of tools, which we call telescopes.
Well, I think we have heard an incredible conversation here from brilliant people who've made unbelievable contributions, moved our science and our planet forward in so many different ways, motivated by curiosity, tenacity, and this sense of the impact that you can have through science. It's magical stuff. I know you're working with other young scientists and you're motivating them in the same way. You believe in the teamwork and the funding. And I think you and we all thank Kavli for recognizing you and your work and what that means and how we model the very best of what we can be and what we can discover. So thank you so much for all that you've done.
Wow, that was impressive. And I was finding it remarkable how we can see how basic research helps us understand our universe. It helps us understand ourselves. And it has a huge impact on curing human diseases and other diseases. So I think this was a great example of how basic research is so important. I want to thank Frank Sesno for being a great moderator. And again, thank Johns Hopkins for hosting us at this beautiful center. And of course, to congratulate again all the laureates who are here today. And just in closing, as you leave, I hope that you will take with you, first of all, an appreciation for the importance of basic science, first and foremost. And that's why the Kavli Foundation supports basic science, what you've heard here today. I hope you also have developed a sense of wonder about our universe. And of course, most importantly, I think that this provides at least me, and I hope all of you, with hope for a very bright future ahead. So thank you very much. I appreciate your coming.